And Dom has been up here at the School of Biomedical Sciences for a few years now, continuing his research program on the molecular control of tissue growth. So Dom's going to talk a little bit about mammalian forebrain development today, and his title is Centrosomal Mechanisms in Mammalian Forebrain Development. Dom, over to you, mate.
Thanks very much, Mike. So let me just share my screen. Okay, so hopefully you can all see that sing out if you can't. So thanks for that introduction, Mike. I'll start actually by confessing a major case of imposter syndrome. My background is not in neuroscience or in developmental biology, but rather biochemistry and cell biology. So I really appreciate the invitation to speak in this forum today. And the work I'll be talking about, which is work characterizing this junk-associated, centrosome-associated microcephaly protein, WD repeat protein 62 or WDR62, how it's involved in brain growth. This work wouldn't be possible with the help of excellent collaborators. Firstly with Julian, back in the day when we were both in Melbourne, and more recently with Mike, my next door neighbor here at UQ. And while I'm acknowledging people, I'll also quickly acknowledge the PhD students that have done this work over the years. In my lab, Yvonne, Nicholas, and more recently, Balazs Shahayab, who just recently graduated and is now a postdoctoral fellow in Helen Cooper's lab, who I think was in the audience, but she might have dropped off. So, I'll start at the beginning, actually, on how we got into this work. We were interested, actually, in these C-Jun N-terminal kinases, or junk kinases, and how this junk pathway was regulated. Now, junk is a member of the mitogen-activated protein kinase family, or the MAP kinase family. And this particular kinase family is characterized by this sort of activation by cascading kinases. And they constitute a major signaling node in the cell because they integrate multiple signals from upstream activators, from a variety of different stimuli, and then can target a broad range of substrates to regulate many different biological outcomes. And you may be familiar with the junk pathway because it's critical for early brain morphogenesis and for neurotube closure, as you can see here. Now, I've said here that that junk signaling is like the Batman villain Two-Face, and if you're not a massive nerd like me, Two-Face is a villain in the DC comic universe, seen here played by Aaron Eckhart in the movie The Dark Knight Returns. And Two-Face is a criminally insane villain, right? And he's obsessed with this concept of duality and the conflict between good and evil or good and bad, and often makes decisions by flipping a coin. So there's this similar feature of duality in junk signaling, which remains enigmatic, particularly in the brain where the consequences of junk activation can be often diametrically opposed. Right? So to give you a few examples, junk signaling is historically considered a pro-apoptotic factor in neurons, and junk is considered a target for things like stroke injury, trauma, various neurodegenerative conditions. But that ignores, I think, abundant evidence that junk also promotes cell survival and proliferation. As another example, junk promotes neurite outgrowth, axon outgrowth, and guidance. But if you sever an axon, the retraction of that axon also involves or is mediated by junk activation. And there's many examples like this. Junk activation can be both pro and anti-inflammatory. In some contexts, it's oncogenic, and in other contexts, it's tumor suppressive. So we're really interested in this sort of the molecular basis for this sort of duality in junk function. Now, you might argue it's to do with cell or stage-specific expression or something to do with isoform selectivity. There's 3 different isoforms of junk in the brain. And you'd be partly right, but these explanations are not wholly satisfactory in that they do not fully account for the functional complexity of the pathway, not by a long shot, actually. So, and junk certainly doesn't flip a coin like Two-Face. It's not random. It's certainly regulated in some way. So one of the things we were working on was this idea that a particular junk molecule could be scaffolded together with its upstream activators into a signaling module or complex. And in this way, its activity could be spatially and temporally compartmentalized to target a specific substrate or a set of substrates. And in this way, you could have 2 different junk molecules performing essentially different functions within the same neuron because they would be scaffolded together by different interacting proteins. So I set out looking for novel junk interacting proteins by performing a yeast-2 hybrid screen, which is a technique no one really does anymore, so I won't go into detail except to say it uses yeast mating and the transcriptional expression of a selectable marker to identify positive interactors between our bait protein, which is junk1 in this case, and novel interactors from a library of potential binders. And what we found was, as the most frequent interactor in the screen was this protein called WDR62, which at the time was completely uncharacterized and had no known function. What was particularly interesting about WDR62 was that it demonstrated selectivity for junk isoforms and binds junk1 preferentially. And it displays very tightly coordinated subcellular localization that was dependent on cell cycle stage. So in mitosis, for example, you can see it's recruited to the poles of the mitotic spindle and surround the centrosomes, which are labeled here in red. You can see WDR62 labeled in green, surrounding the centrosomes during these mitotic stages, indicating a role in spindle and mitotic regulation. In non-mitotic cells, in ciliated cells, for example, it's also localized to the centrosomes, or which is also known as the basal body of the primary cilium, as you can see here. So we were really excited by this, as it indicated what we had found was a protein that delineated sort of widely reported junk functions in cytoskeletal or cell cycle regulation, as distinct from, say, its functions in regulating programmed cell death, which might be mediated at the mitochondria, for example. And just by way of some background, if you're not familiar, centrosomes are these protein-dense, membraneless organelles that are formed by protein-protein interactions, essentially. So at its core are these barrels of atypical tubulin known as centrioles, and these centrioles duplicate using a template mechanism. So in other words, you need a mother centriole to make a daughter. And these mother and daughter centrioles split during cell division so that each daughter cell inherits a copy of the centrosomes, much like it inherits a copy of DNA. Centrosomes are also known as microtubule organizing centers because they anchor and nucleate the formation of microtubules to coordinate the microtubule cytoskeleton. And depending on cell cycle stage, this is involved in, for example, the growth of ciliary axonemes, which are basically bundles of microtubules to make primary and motile cilia. In mitosis, they participate in spindle formation, and of particular relevance to this talk also connects the bipolar spindle to the plasma membrane, and in this way positions the spindle axis within three-dimensional space and time, and this ultimately determines the division plane or the cleavage plane to orient mitotic divisions. Centrosomes are also known as signaling hubs because their protein-dense structures localize many different kinases and phosphatases that are involved in cell cycle regulation or DNA damage responses. Now, coming back to WDR62, it was also around this time, about 10 years ago, when that WDR62 was identified by several groups, including groups led by Murat Günel, Chris Walsh, and Jeff Woods, as mutated in the MCPH2 locus, and is the second most common cause of primary microcephaly in humans, accounting for about 15% of cases. Now, this made a lot of sense, actually, because many microcephaly genes, the majority of microcephaly genes, encode centrosomal components. As you can see here, this list is a little bit out of date. I think we're up to about 17 microcephaly genes now. And I'm sure we'll hear about a few more of these from Louisa's talk. But what you can see is that centrosomal proteins are sort of overrepresented in these sorts of lists, which suggests that centrosomal functions in neurostem and progenitor cells are critical for brain growth. And over the years, 40 different mutations of WDR62 have been identified that result in protein loss in patients due to severe truncation or altered splicing of the mRNA, leading to nonsense-mediated decay. And you can also see here that WDR62 mutations are associated with a broad spectrum of cortical malformations, which suggests multiple functions in neurodevelopment. So Julian helped us out with our first experiments looking at the role of WDR62 in embryonic brain growth using in utero electroporation to deplete the protein with siRNA, and by staining with proliferation markers and by doing BRD pulse labeling experiments, we showed that WDR62 loss led to a reduced proliferation and increased cell cycle exit of radioglial cells, which, as you know, are the neurostem cells in the subventricular zone of the developing cortex. In subsequent studies not shown here, we also showed that this reduction in radioglial cell numbers coincided with an increase in the production of intermediate progenitors and immature neurons. So there was a process of premature or precocious differentiation that occurred when you knocked out WDR62. So WDR62 loss had a detrimental impact on the balance of these sorts of symmetric divisions that expand the pool of neurostem cells and asymmetric divisions that lead to neurogenesis. These neurodevelopmental deficits were phenocopied by depletion of the JNK1 isoform specifically, but not the JNK2 isoform. You can see this here, that JNK1 and WDR62 depletion by siRNA using in utero electroporation sort of mimicked one another, which sort of indicates that WDR62 and JNK1 works within the same pathway. And this is consistent with our biochemical studies, which showed isoform selectivity in binding specific JNK isoforms, right? And in support of this, the expression of a constitutively active mutant of JNK1 rescued the decrease in the SOX2 positive cells caused by WDR62 depletion. And as you know, SOX2 delineates radioglial cells from intermediate progenitors, and it also reversed the precocious generation of these immature neurons marked by TUJ1. I'm just noticing that maybe my laser pointer isn't showing up, but oh, hang on. Sorry, my screen is frozen just briefly. Hopefully it'll recover. All right, there we go. So following on from those in utero electroporation studies, we then used CRISPR-Cas9 to generate knockout animals to reveal the phenotype. And at the same time, we also generated mice with knock-ins of a couple of missense mutations commonly identified in patients. Now, patients with some of these missense mutations retain WDR62 protein expression, which is interesting. And these missense mutations impact evolutionary conserved amino acids in the WD repeat regions of the protein, which are thought to be involved in protein-protein interactions. So we reason that these animals could be informative from a sort of mechanistic point of view. So what we found with these animals that were homozygous for these mutations was that they variably expressed a phenotype of reduced embryonic growth and maldevelopment of one or both eyes, and perished prior to birth. But then a proportion of homozygous animals do survive to postnatal stages, and these were by and large phenotypically normal, with the exception that they present with smaller brains in terms of size and volume, and they have reduced cortical thickness, consistent with the role for WDR62 in neural proliferation. And during embryonic development around E12.5 and 14.5, we found that radioglial cell numbers, we found reduced radioglial cell numbers and the precocious differentiation into intermediate progenitors and neurons, consistent with what we'd observed with the in utero electroporation studies. The other universal phenotype amongst these WDR62 mutant animals was that they were invariably infertile, indicating important functions in gametogenesis and in meiotic divisions. And I'll also note here that infertility is in general sort of commonly observed following knockout of these centrosome-associated microcephaly genes. So I just want to quickly recap as I've gone to that, well, sort of fairly quickly. WDR62 is encoded by the MCPH2 locus, and mutations cause a broad clinical phenotype of brain malformation. It's a JNK1 interacting protein that's localized to spindle microtubules, And to centrosomes, they may delineate junk1 functions at these subcellular locales. And importantly, it's required to to maintain neurostem cell numbers and the growth of the neocortex consistent with defects in these functions underlying the clinical condition, right, of microcephaly. So we next asked the question, well, what exactly does WDR62 do in radioglial cells, and how does it determine brain size? And whilst its localization to centrosomes gives us a clue, centrosome functions in neurostem cell biology is actually quite diverse, as you can see here, and defects in one, some, or all of these things may represent pathogenic mechanisms in microcephaly. So this includes things such as cilia formation, so radioglial cells at the apical surface project a primary cilium into the ventricular space, and this acts as like a sensory appendage for morphogens and neurotrophic factors and hormones and whatnot. Centrosomes also regulate cell cycle progression, and the length of time taken to complete a cell cycle is important if an organism needs to fit in, say, a certain number of cell replications within the developmental window for normal tissue growth, right? And as I've alluded to, centrosomes also regulate spindle formation and spindle orientation, and this impacts division symmetry, depending on the geometry of radioglial cell divisions relative to the apical surface. And here, whilst daughter cells each inherit a centriole, these are biochemically and potentially functionally distinct, as one centriole is a whole cell cycle older than the other centriole, the daughter. And this asymmetry in centriole inheritance may also determine daughter cell fate, particularly if fate determinants are biased towards associating with one centriole over the other. And lastly, centrosomes also determine cell polarity, which may be significant, for example, in the migration of these immature neurons along glial projections, which is required to layer the cortex. So we looked more closely at centrosomes and microtubules in live cell imaging studies, and hopefully this plays without too much issue. Okay. So these are 293 cells, and what we found with either WDR62 depletion with siRNA or deletion using CRISPR, what we found was that this resulted in reduced cohesion of the spindle poles. So hopefully you can see that if you look closely here at the poles of this spindle in these knockout cells, you see a reduction in integrity of those poles. And whilst control cells establish a spindle polarity and divide in a timely manner, hopefully you can appreciate here in these knockout cells that these spindles undergo considerable movement and rotation. And this was accompanied by prolonged mitoses. And missense mutations, when we looked at them, the ones that we were looking at also disrupted the localization of WDR62 to the spindle pole, which suggests that abnormalities in spindle functions may be linked to the microcephaly phenotype. So this function in maintaining division orientation is conserved in neurostem cells in vivo, whether we looked at Drosophila larval neuroblasts, which divide along a strict apical basal axis, or in radioglial cells dividing at the apical ventricular surface. And in both of these model systems, you can see that either WDR62 depletion or knockout or missense mutation significantly disrupts spindle orientation. And this impacts division symmetry. And we believe this then causes abnormalities in neurostem cell proliferative versus neurogenic divisions, leading to a deficit in brain growth. Now, because we observed the same defect across different animal models and with different WDR62 mutations, this gives us some confidence that these specific molecular and cellular functions that we'd identified here were responsible for the neural phenotypes that we observed, right? But to prove that conclusively, we also needed to work out how WDR62 was recruited to the spindles in order to specifically disrupt that process, right? And to do that, we went fishing again, this time using WDR62 as the bait, but using a proteomic technique known as proximity bioaccumulation coupled with mass spectrometry, essentially a fancier rod and reel. And basically what we did was we generated a fusion of WDR62 with a promiscuous biotin ligase, that's this BRA enzyme, and expressed this in cells. And by feeding biotin in a specific context, for example, mitotic versus non-mitotic, our bait protein would then be able to biotinylate surrounding proteins that are in close proximity to our bait in a cell cycle stage specific fashion. You could then isolate these biotinylated proteins using affinity chromatography and identify them using the mass spectrometer. And by doing this, we found things that we would expect. So things like, you can see JNK1 on this list here, tubulin, and a mitotic kinase called Aurora A, which signals the WDR62, and this is work I haven't had time to go into. But these are basically interactors that we know about, and we were pleased to see that they were identified in this list. But our top hits in terms of the numbers of peptides was the centrosomal protein called CEP170. And in preliminary work, we confirmed the interaction between WDR62 and CEP170 by co-IP, and showed that CEP170 phenocopies the spindle defects when you knock it down using siRNA. So we think that this interaction is likely involved in spindle regulation, right, or working together in spindle regulation. Now, CEP170 is particularly interesting because it's localized to these distal appendages, and so are present specifically on mother or matured mother centrioles. And here you can see this in cells at late G2 when the centrosomes have duplicated. You can see that CEP170 is only localized to one of these centrosomes, the mother, and not the daughter. So this is very interesting because not only do we think that CEP170 recruits WDR62 to the centrosomes, but during early mitosis, as the spindle starts to form, what you have here is the potential for biased recruitment of WDR62 and JNK1 to one pole of the spindle, but not the other. And you can see this here in the cell in early prophase. You can see that WDR62 and CEP170 is co-localized on one centrosome, but not the other, right? So if this starts to stabilize the microtubules on one pole, but not the other, this is one mechanism on how you can get bias attachment of the spindle towards the plasma membrane. So for example, the side closest to the apical surface to precisely orient that spindle and to position the mother centriole in exactly the same way with each stem cell division. So this work's not finished. We're still working on this, but this is currently the model that we're working towards. What we think is happening is that upon a transition into mitosis, the mitotic kinase Aurora A, which is upregulated at this time, phosphorylates WDR62 to promote its interaction with CEP170 and bias the recruitment of WDR62 to the older or more mature centrosome. WDR62 and JNK would then stabilize microtubules and the attachment of this mother centrosome to the plasma membrane and other extrinsic cues in order to establish spindle polarity. And because daughter centrioles mature and develop these distal appendages as the cell progresses through mitosis, WDR62 will eventually be recruited to stabilize the opposite pole, but this pole would now be located away from the apical surface, for example. And this axis orients radioglial divisions, we think, in order to determine symmetric versus asymmetric divisions. So this is my last slide, and I only have this here to reinforce the fact that the story is far from complete. The significance of characterizing these WDR62 mechanisms in detail is so that we can delineate its functions in spindle regulation as distinct from other things that might also contribute to brain growth. So for example, I previously shown that WDR62 is localized to cilia and to the basal body of the primary cilium, and in work I haven't had time to go into. We've also shown that WDR62 is required for the extension of the ciliary axoneme, marked here by IL-13B in green. You can see here that WDR62 knockout or mutations leads to cilia formation defects in radioglial cells in vivo. And importantly, this cilia function doesn't involve CEP170, but actually another microcephaly protein called CEMPJ, which I won't go into. Okay, but basically by delineating these protein interaction networks involving WDR62, this will allow us to define consequences of these signaling interactions. So for example, we can then target the WDR62 CEP170 interaction in neurostem cells rather than the protein components themselves in order to reveal their neurodevelopmental consequences and a specific function for centrosomes, such as in oriented cell division. So I'll stop there and thank you very much for listening, ramble on for much too long, I'm sure, and I'm happy to take questions.
Thanks, Dom. Beautiful work as always. So there's a question from Julian on the chat. Not sure if this is relevant, but is the pathological action of R439H substitution variant of WDR62 explained in part by ectopic interactions with non-native proteins compared to the wild type?
Yeah, so this is an interesting question, whether this particular missense mutant, which is still expressed in mice and in humans, whether it's a loss of function mutation, whether it's actually a gain of function mutation by, it's there, it's disrupting, it's an expression of a mutant protein that can't do its normal function, so that it's there to disrupt some sort of process. It's possible, but we haven't found any evidence of that yet, Julian. So for example, if that were the case, you would imagine that the heterozygous animals would also present with some kind of phenotype. And so far, we haven't found anything. That particular animal, the heterozygous animals seem perfectly normal. Maybe we're not looking closely enough, and we certainly haven't spent as much time on that particular genotype of animals than the others, but so far we haven't seen anything overtly defective. So, yeah, don't have a clear answer for you, unfortunately.
Dom, with that WDR62 CEP170 axis potentially contributing to microcephaly, do you think they're also interacting to regulate development, for instance, of the testis, if you've got infertility in these mice?
It's potentially the case. So in these germ stem cells, WDR62 is localized to the spindle poles of them, and they do impact the orientation of meiotic as well as mitotic divisions of these sorts of germ stem cells. And that's been published actually by another group. They also similarly implicate CEP170 in this sort of process. The infertility is a combination of multiple things, though, that being one thing, but also the infertility, for example, in sperm is also due to defects in basal body functions. So those even when you can make a spermatocyte, those things can't swim properly because they can't make a proper tail. So there's a combination of different defects that lead to infertility, and I think that's why it's sort of all the animals are infertile, whereas some of the other phenotypes can be sort of overcome.